Current classification of bacteria. On the basis of 16 sRNA sequences, it is now clear that there are at least 3 domains of organisms, the 2 prokaryotic groups, Archaebacteria and Eubacteria, and Eukarya figure 1. Within the Eubacteria there are at least 11 groups, referred to sometimes as phyla, which can be represented on a phylogenetic tree figure 2. What can be seen is the wide diversity within the bacterial world, and that many of the bacteria that may be familiar to us, such as E. coli, Bacillus species, and Neisseria gonorrhoeae, are located within just a few of these phyla. It should be noted that the number of phyla will increase as more bacteria from diverse habitats are sequenced. Tissues, organs, and organ systems. Cells are the basic building blocks of multicellular animals. When cells with the same characteristics or specializations are grouped together, they form a tissue. There are 4 basic types of tissues: epithelial, connective, muscle, and nervous, but there are variations on each basic type. An organ is usually made up of several different tissue types. Figure 1. Epithelial tissues cover the body and line organs. Epithelial tissues are sheets of densely packed, tightly connected cells that cover inner and outer body surfaces. They form the skin and line hollow organs of the body, such as the gut. Figure 2. Some epithelial cells have secretory functions. Examples are the groups of epithelial cells that secrete hormones, milk, mucus, digestive enzymes, or sweat. Other epithelial cells have cilia to help substances move over surfaces or through tubes. Since epithelial cells create boundaries between the inside and the outside of the body and between body compartments, they frequently have protective as well as absorptive and transport functions. Epithelial cells can also form receptors that provide information to the nervous system. Smell and taste receptors, for example, are epithelial cells that detect specific chemicals. Epithelial tissues have distinct inner and outer surfaces. The outer surface faces the air, as in the case of the skin and lungs, or a fluid-filled organ cavity, such as the lumen of the gut. These outer surfaces are the apical ends of the epithelial cells, which may have cilia or may be highly folded to increase their surface area. Classification of bacteria. The main aim of bacterial taxonomy is to set up a classification and ordering of bacteria into groups based on common properties. This classification can then be used to identify individual bacterial species or strains, identification, and name of nomenclature. Classification is also used to study relationships of different bacterial groups based on shared phenotypic properties and probably a common evolutionary history. A wide range of morphological, biochemical, and molecular characters are measured, and the information derived from this is used to create a hierarchical classification system into which individual bacteria can be placed. The most important rank in the taxonomic hierarchy is called the species, which from the time of Linnaeus 1707-1778 have been named in a similar way. Related species are grouped into a genus place. Genera, which is usually based around some distinct phenotypic character, and the generic name is used as part of a binomial species name. So, for example, Escherichia coli is in the genus Escherichia named after Escherick, who first isolated the bacteria in 1885. The specific epithet, coli, is in this case an indication as to where the organism was isolated from, i.e. the colon. However, note that the binomial name is a label, not a description. It does not have to describe the species in any way. Note too that the genus name has a capital initial letter, but the specific epithet, which cannot be used alone, does not, and that the whole binomial name is Latinized and therefore should be italicized or underlined. Normally, once the name of the organism has been written in full, the genus name can be reduced to a letter or letters as long as there can be no confusion with other genera. Thus, Escherichia coli will subsequently be written as E. coli. The higher taxonomic categories into which the organism can be ranked are sequentially, family, order, class, division, and kingdom, though the final 3 are not particularly useful in bacteriology. Similarly, species may be divided into subspecies, and a culture of a particular organism is usually referred to as a strain. Strain is also used to describe a subgroup of a species that shares some features such as one type of O-antigen or mutation. There are also informal names, not italicized, which are sometimes useful for describing groups of organisms such as the green sulfur bacteria or the spirochetes. The main impetus for bacterial classification has been the need to be able to identify bacterial disease-causing organisms, and therefore these microbes are particularly well classified. However, it should be remembered that many other species have not been well characterized and many have not been isolated or described at all. The authority on bacterial classification is Berthi's Manual of Systematic Bacteriology, which contains the nearest possible complete listings of all prokaryotic species and their distinguishing characteristics. Characters used in bacterial identification. A list of the types of characters used to distinguish between bacteria is given below. This is not an exhaustive list, and equally not all characters are used in either the classification or identification of every bacterial species. In many cases of well-known microorganisms, a small set of tests may be all that is required to identify a microbe positively, especially if the site from which it has been isolated and the disease it caused are known. A comma-shaped, gram-negative rod isolated from a patient excreting copious amounts of straw colored, liquid stools may be enough to identify it putatively as Vibrio cholera, the causative agent for cholera. However, it may be necessary to do a wide range of tests to determine the name of an isolated if it is an undescribed microbe. A. Colony morphology. The shape, texture, and color of bacterial colonies can be distinctive. For example, Staphylococcus aureus is so named as colonies are of a yellow color. Aureus comes from the Latin for golden. B. Cell shape, structures, and reaction to stains. Whether bacteria are gram-positive or gram-negative is one of the most important clinical diagnostic tests. Gram staining and microscopic examination of bacteria deformation as to the shape, size, and arrangement of the bacterial cells, and in some cases this is enough to identify certain genera such as streptococci, which are gram-positive cocci that grow in chains. Stains may also be used to show other morphological features, which may be used for classification, such as the presence of spores, malachite green stain, unusual cell walls, Zeal-Nielsen acid fast stain, capsules, Indian ink stain, intercellular lipids, Sudan black flagellar stain, metachromatic granules, Albert Laybourne stain. C. Growth characteristics, the temperature, pH, and O2 requirements of isolate are useful tests for the identification of bacteria. D. Biochemical tests. There is a wide range of tests available that measure various aspects of bacterial metabolism. What carbon and nitrogen sources the bacteria can use. The end products of their metabolic processes, such as acetylcholine, tested for in the Vieux Proskauer test. What enzymes the bacteria produce, such as decarboxylases, proteases, and denases. The presence of other molecules such as toxins, long-chain fatty acids, or antibiotics. E. Immunological tests. Antibodies to cellular components such as the O-side chains of lipopolysaccharide (LPS) or capsules are frequently used to distinguish between strains of one species. F. DNA tests. Comparisons of DNA content and sequence between strains are the most definitive methods for separating organisms into different groups and for measuring the evolutionary relatedness between them. Phylogenetic analysis. Traditionally, DNA was analyzed by measuring the GC content or the amount of DNA homology between strains. More recently, the comparison of the actual DNA sequences of conserved cellular molecules such as 16S ribosomal RNA or proteins such as cytochrome C, APase, and elongation factor has been used. This information has been analyzed by a technique called cladistics to trace through phylogenetic lineages that have not been obscured by patterns of convergent evolution. Current classification of bacteria. On the basis of 16 sRNA sequences, it is now clear that there are at least 3 domains of organisms, the 2 prokaryotic groups, Archaebacteria and Eubacteria, and Eukarya figure 1. Within the Eubacteria there are at least 11 groups, referred to sometimes as phyla, which can be represented on a phylogenetic tree figure 2. What can be seen is the wide diversity within the bacterial world, and that many of the bacteria that may be familiar to us such as E. coli, Bacillus species, and Neisseria gonorrhoeae are located within just a few of these phyla. It should be noted that the number of phyla will increase as more bacteria from diverse habitats are sequenced. Tissues, organs, and organ systems. Cells are the basic building blocks of multicellular animals. When cells with the same characteristics or specializations are grouped together, they form a tissue. There are 4 basic types of tissues: epithelial, connective, muscle, and nervous, but there are variations on each basic type. An organ is usually made up of several different tissue types. Figure 1. Epithelial tissues cover the body and line organs. Epithelial tissues are sheets of densely packed, tightly connected cells that cover inner and outer body surfaces. They form the skin and line hollow organs of the body, such as the gut. Figure 2. Some epithelial cells have secretory functions. Examples are the groups of epithelial cells that secrete hormones, milk, mucus, digestive enzymes, or sweat. Other epithelial cells have cilia to help substances move over surfaces or through tubes. Since epithelial cells create boundaries between the inside and the outside of the body and between body compartments, they frequently have protective as well as absorptive and transport functions. Epithelial cells can also form receptors that provide information to the nervous system. Smell and taste receptors, for example, are epithelial cells that detect specific chemicals. Epithelial tissues have distinct inner and outer surfaces. The outer surface faces the air, as in the case of the skin and lungs, or a fluid-filled organ cavity, such as the lumen of the gut. These outer surfaces are the apical ends of the epithelial cells, which may have cilia or may be highly folded to increase their surface area. The inner surfaces of an epithelium are the basal ends of the epithelial cells, which rest on an extracellular matrix called the basal lamina. The skin and the lining are the better examples of epithelial tissues that receive much wear and tear. Accordingly, cells in these tissues have a high rate of cell division to replace cells that die and are shed. Dandruff consists of discarded skin cells. Connective tissues support and reinforce other tissues. In contrast to densely packed epithelial tissues, connective tissues consist of dispersed populations of cells embedded in an extracellular matrix that they secrete. The composition and properties of the matrix differ among types of connective tissues. An important component of the extracellular Accordingly, cells in these tissues have a high rate of cell division to replace cells that die and are shed. Dandruff consists of discarded skin cells. Connective tissues support and reinforce other tissues. In contrast to densely packed epithelial tissues, connective tissues consist of dispersed populations of cells embedded in an extracellular matrix that they secrete. The composition and properties of the matrix differ among types of connective tissues. An important component of the extracellular matrix secreted by connective tissue cells is protein fibers. The dominant protein in the extracellular matrix is collagen. See figure 4.26. Collagen is, in fact, the most abundant protein in the human body, representing 25% of total body protein. Collagen fibers are strong. They give the connective tissue of skin, tendons, and ligaments resistance to stretch. Similarly, collagen fibers provide a net-like framework for organs, giving them shape and structural strength. Connective tissue that fills spaces between organs has a low density of collagen fibers. Another type of protein fiber in the extracellular matrix of connective tissues is the stretchable protein elastin. It can be stretched to several times its resting length and then recoil. Fibers composed of elastin are most abundant in tissues that are regularly stretched, such as the walls of the lungs and the large arteries. Gradual loss of elastin fibers with age causes gradual loss of resiliency of the skin. Cartilage and bone are connective tissues that provide rigid structural support. In cartilage, a network of collagen fibers is embedded in a flexible matrix consisting of a protein-carbohydrate complex. Cartilage, which lines the joints of vertebrates, is resistant to compressive forces. Since it is flexible, it provides structural support for flexible structures such as external ears and noses. The extracellular matrix in bone also contains many collagen fibers, but it is hardened by the deposition of the mineral calcium phosphate. Adipose tissue is a form of loose connective tissue that includes adipose cells, which form and store droplets of lipids. Adipose tissue, or fat, is a major source of stored energy. It also serves to cushion organs, and layers of adipose tissue under the skin can provide a barrier to heat loss. Blood is a connective tissue consisting of cells dispersed in an extensive extracellular matrix, the blood plasma. The blood plasma is much more liquid than the extracellular matrices of the other connective tissues, but it too contains an abundance of proteins. Muscle tissues contract. Muscle tissues consist of elongated cells that can contract and cause movement. Muscle tissues are the most abundant tissues in the body, and when animals are active, they use most of the energy produced in the body. Nervous tissues process information. There are two basic cell types in nervous tissues, neurons and glial cells. Neurons, which are extremely diverse in size and form, communicate via electrochemical signals. These nerve impulses can be conducted via long extensions of the neurons to other parts of the body, where they are communicated to other neurons, muscle cells, or secretory cells. Neurons are involved in controlling the activities of most organ systems to achieve homeostasis. Glial cells do not generate or conduct electrochemical signals, but they provide a variety of supporting functions for neurons. There are more glial cells than neurons in our nervous systems. Organs consist of multiple tissues. A discrete structure that carries out a specific function in the body is called an organ. Examples are the stomach, the heart, the liver, and the kidney. Most organs include all four tissue types. The wall of the stomach is a good example. The inner surface of the stomach that contacts food is lined with a sheet of epithelial cells. Some of the epithelial cells secrete mucus, enzymes, or stomach acid. Beneath the epithelial lining is connective tissue. Within this connective tissue are nerves, glands, clusters of secretory epithelial cells, and blood vessels. Concentric layers of smooth muscle tissue enable the stomach to contract to mix food with digestive juices. A network of neurons between the muscle layers controls these movements and also influences the secretions of the stomach. Surrounding the stomach is a sheath of connective tissue. An individual organ is usually part of an organ system, a group of organs that function together. The stomach is part of the digestive system, which also includes the food tube, esophagus, the small and large intestines, the pancreas, which secretes digestive enzymes, and the liver, which secretes bile. The major organ systems of mammals are outlined in Table 1. Bacteria and archaea, the prokaryotic domains. The organisms most commonly found in such extremely acidic environments belong to the two major groups of prokaryotes, bacteria and archaea. The bacteria live in almost every environment on Earth. The archaea are a superficially similar group of microscopic, unicellular prokaryotes. However, both the biochemistry and the genetics of bacteria differ in numerous ways from those of archaea. Not until the 1970s did biologists discover how radically different bacteria and archaea really are. And only with the sequencing of an archaeal genome in 1996 did we realize just how extensively archaea differ from both bacteria and eukaryotes. Many biologists acknowledge the antiquity of these clades and the importance of their differences by recognizing three domains of living things: bacteria, archaea, and eukarya. The domain bacteria comprises the true bacteria. The domain archaea, Greek archaios, ancient, comprises other prokaryotes once called inaccurately ancient bacteria. The domain eukarya includes all other living things on Earth. Dividing the living world in this way, with two prokaryotic domains and a single domain for all the eukaryotes, fits with the current trend toward reflecting evolutionary relationships in classification systems. In the 8 chapters of Part 5, we celebrate and describe the diversity of the living world, the products of evolution. This chapter focuses on the two prokaryotic domains. Why three domains? What does it mean to be different? You and the person nearest you look very different. Certainly you appear more different than the two cells. But the two of you are members of the same species, while these two tiny organisms that look so much alike actually are classified in entirely separate domains. Still, all three of you in the domain eukarya and those two prokaryotes in the domains bacteria and archaea have a lot in common. Members of all three domains. Conduct glycolysis. Replicate their DNA semi-conservatively. Have DNA that encodes polypeptides. Produce these polypeptides by transcription and translation and use the same genetic code. Have plasma membranes and ribosomes in abundance. There are also major differences among the domains. Members of the eukarya have cells with nuclei, membrane-enclosed organelles, and a cytoskeleton, structures that no prokaryote has. And a glance at Table 1 will show you that there are also major differences, most of which cannot be seen even under the microscope, between the two prokaryotic domains. In some ways the archaea are more like us, in other ways they are more like bacteria. Genetic studies have led many biologists to conclude that all three domains had a single common ancestor, and that the present-day archaea share a more recent common ancestor with eukaryotes than they do with bacteria. Figure 1. Because of the ancient time at which these three clades diverged, the major differences among the three kinds of organisms, and especially the likelihood that the archaea are more closely related to the eukaryotes than are either of those groups to the bacteria, many biologists agree that it makes sense to treat these three groups as domains, a higher taxonomic category than kingdoms. To treat all the prokaryotes as a single kingdom within a 5-kingdom classification of organisms would result in a kingdom that is paraphyletic. That is, a single kingdom, prokaryotes, would not include all the descendants of their common ancestor. The common ancestor of all three domains was prokaryotic. Its genetic material was DNA, its machinery for transcription and translation produced RNAs and proteins, respectively. It probably had a circular chromosome, and many of its structural genes were grouped into operons. The archaea, bacteria, and eukarya of today are all products of billions of years of natural selection and genetic drift, and they are all well adapted to present-day environments. None are primitive. The common ancestor of the archaea and the eukarya probably lived more than 2 billion years ago, and the common ancestor of the archaea, the eukarya, and the bacteria probably lived more than 3 billion years ago. The earliest prokaryotic fossils date back at least 3.5 billion years, and these ancient fossils indicate that there was considerable diversity among the prokaryotes even during the earliest days of life. The prokaryotes were alone on Earth for a very long time, adapting to new environments and to changes in existing environments. They have survived to this day, and in massive numbers. General biology of the prokaryotes. There are many, many prokaryotes around us, everywhere. Although most are so small that we cannot see them with the naked eye, the prokaryotes are the most successful of all creatures on Earth, if success is measured by numbers of individuals. The bacteria in one person's intestinal tract, for example, outnumber all the humans who have ever lived, and even the total number of human cells in their host body. Some of these bacteria form a thick lining along the intestinal wall. Bacteria and archaea in the oceans number more than 3 times 1028. This stunning number is perhaps 100 million times as great as the number of stars in the visible universe. Although small, prokaryotes play many critical roles in the biosphere, interacting in one way or another with every other living thing. In this section, we'll see that some prokaryotes perform key steps in the cycling of nitrogen, sulfur, and carbon. Other prokaryotes trap energy from the sun or from inorganic chemical sources, and some help animals digest their food. The members of the two prokaryotic domains outdo all other groups in metabolic diversity. Eukaryotes, in contrast, are much more diverse in size and shape, but their metabolism is much less diverse. In fact, much of the energy metabolism of eukaryotes is carried out in organelles, mitochondria, and chloroplasts that are descended from bacteria. Prokaryotes are found in every conceivable habitat on the planet, from the coldest to the hottest, from the most acidic to the most alkaline, and to the saltiest. Some live where oxygen is abundant and others where there is no oxygen at all. They have established themselves at the bottom of the seas, in rocks more than 2 kilometers into Earth's solid crust, and inside other organisms, large and small. Their effects on our environment are diverse and profound. What do these tiny but widespread organisms look like? Prokaryotes and their associations take a few characteristic forms. Three shapes are particularly common among the prokaryotes: spheres, rods, and curved or spiral forms. Figure. A spherical prokaryote is called a coccus plural, cocci. Cocci may live singly or may associate in 2 or 3-dimensional arrays as chains, plates, blocks, or clusters of cells. A rod-shaped prokaryote is called a bacillus plural, bacilli. Spiral forms are the third main prokaryotic shape. Bacilli and spiral forms may be single or may form chains. Prokaryotes are almost all unicellular, although some multicellular ones are known. Associations such as chains do not signify multicellularity because each cell is fully viable and independent. These associations arise as cells adhere to one another after reproducing by fission. Associations in the form of chains are called filaments. Some filaments become enclosed within delicate tubular sheets. Prokaryotes lack nuclei, organelles, and a cytoskeleton. The basic unit of archaea in bacteria is the prokaryotic cell, which contains a full complement of genetic and protein synthesizing systems, including DNA, RNA, and all the enzymes needed to transcribe and translate the genetic information into proteins. The prokaryotic cell also contains at least one system for generating the ATP it needs. In what follows, bear in mind that most of what we know about the structure of prokaryotes comes from studies of bacteria. We still know relatively little about the diversity of archaea, although the pace of research on archaea is accelerating. The prokaryotic cell differs from the eukaryotic cell in 3 important ways. First, the organization and replication of the genetic material differs. The DNA of the prokaryotic cell is not organized within a membrane-enclosed nucleus. DNA molecules in prokaryotes, both bacteria and archaea, are usually circular. In the best studied prokaryotes, there is a single chromosome, but there are often plasmids as well. Second, prokaryotes have none of the membrane-enclosed cytoplasmic organelles that modern eukaryotes have: mitochondria, Golgi apparatus, and others. However, the cytoplasm of a prokaryotic cell may contain a variety of infoldings of the plasma membrane and photosynthetic membrane systems not found in eukaryotes. Third, prokaryotic cells lack a cytoskeleton, and, without the cytoskeletal proteins, they lack mitosis. Prokaryotic cells divide by their own elaborate method, fission, after replicating their DNA. Prokaryotes have distinctive modes of locomotion. Although many prokaryotes cannot move, others are mobile. These organisms move by one of several means. Some spiral bacteria, called spirochetes, use a corkscrew-like motion made possible by modified flagella, called axial filaments, running along the axis of the cell beneath the outer membrane. Many cyanobacteria and a few other bacteria use various poorly understood gliding mechanisms, including rolling. Various aquatic prokaryotes, including some cyanobacteria, can move slowly up and down in the water by adjusting the amount of gas in gas vesicles. By far the most common type of locomotion in prokaryotes, however, is it driven by flagella. Bacterial flagella are slender filaments that extend singly or in tufts from one or both ends of the cell or are randomly distributed all around it. Figure. A bacterial flagellum consists of a single fibril made of the protein flagellin, projecting from the cell surface, plus a hook and basal body responsible for motion. In contrast, the flagellum of eukaryotes is enclosed by the plasma membrane and usually contains a circle of 9 pairs of microtubules surrounding 2 central microtubules, all containing the protein tubulin, along with many other associated proteins. The prokaryotic flagellum rotates about its base, rather than being as a eukaryotic flagellum or cilium does. Prokaryotes have distinctive cell walls. Most prokaryotes have a thick and relatively stiff cell wall. This wall is quite different from the cell walls of plants and algae, which contain cellulose and other polysaccharides, and from those of fungi, which contain chitin. Almost all bacteria have cell walls containing peptidoglycan, a polymer of amino sugars. Archaeal cell walls are of different types, but most contain significant amounts of protein. One group of archaea has pseudopeptidoglycan in its wall, as you have probably already guessed from the prefix pseudo. Pseudopeptidoglycan is similar to, but distinct from, the peptidoglycan of bacteria. Peptidoglycan is a substance unique to bacteria. Its absence from the walls of archaea is a key difference between the two prokaryotic domains. In 1884, Hans Christian Gram, a Danish physician, developed a simple staining process that has lasted into our high-technology era as a useful tool for identifying bacteria. The Gram stain separates most types of bacteria into 2 distinct groups, Gram-positive and Gram-negative, on the basis of their staining. A smear of cells on a microscope slide is soaked in a violet dye and treated with iodine. It is then washed with alcohol and counterstained with saffron and or red dye. Gram-positive bacteria retain the violet dye and appear blue to purple. The alcohol washes the violet stain out of Gram-negative cells. These cells then pick up the saffron and counterstain and appear pink to red. Gram staining characteristics are useful in classifying some kinds of bacteria and are important in determining the identity of bacteria in an unknown sample. For many bacteria, the Gram staining results correlate roughly with the structure of the cell wall. Peptidoglycan forms a thick layer outside the plasma membrane of Gram-positive bacteria. The Gram-negative cell wall usually has only 1/5 as much peptidoglycan, and outside the peptidoglycan layer the cell is surrounded by a second, outer membrane quite distinct in chemical makeup from the plasma membrane. Between the inner plasma and outer membranes of Gram-negative bacteria is a periplasmic space. This space contains enzymes that are important in digesting some materials, transporting others, and detecting chemical gradients in the environment. The consequences of the different features of prokaryotic cell walls are numerous and relate to the disease-causing characteristics of some prokaryotes. Indeed, the cell wall is a favorite target in medical combat against diseases that are caused by prokaryotes because it has no counterpart in eukaryotic cells. Antibiotics such as penicillin and ampicillin, as well as other agents that specifically interfere with the synthesis of peptidoglycan-containing cell walls, tend to have little, if any, effect on the cells of humans and other eukaryotes. Prokaryotes reproduce asexually, but genetic recombination does occur. Prokaryotes reproduce by fission, an asexual process. Recall, however, that there are also processes, transformation, conjugation, and transduction, that allow the exchange of genetic information between some prokaryotes quite apart from either sex or reproduction. Some prokaryotes multiply very rapidly. One of the fastest is the bacterium Escherichia coli, which under optimal conditions has a generation time of about 20 minutes. The shortest known prokaryote generation times are about 10 minutes. Generation times of 1 to 3 hours are common for others, some extend to days. Bacteria living deep in Earth's crust may suspend their growth for more than a century without dividing and then multiply for a few days before suspending growth again. What kinds of metabolism support such a diversity of growth rates? Prokaryotes have exploited many metabolic possibilities. The long evolutionary history of the bacteria and archaea, during which they have had time to explore a wide variety of habitats, has led to the extraordinary diversity of their metabolic lifestyles, their use or non-use of oxygen, their energy sources, their sources of carbon atoms, and the materials they release as waste products. Anaerobic versus aerobic metabolism. Some prokaryotes can live only by anaerobic metabolism because molecular oxygen is poisonous to them. These oxygen-sensitive organisms are called obligate anaerobes. Other prokaryotes can shift their metabolism between anaerobic and aerobic modes and thus are called facultative anaerobes. Many facultative anaerobes alternate between anaerobic metabolism, such as fermentation and cellular respiration as conditions dictate. Aerotolerant anaerobes cannot conduct cellular respiration, but are not damaged by oxygen when it is present. At the other extreme from the obligate anaerobes, some prokaryotes are obligate aerobes, unable to survive for extended periods in the absence of oxygen. They require oxygen for cellular respiration. Nutritional categories. Biologists recognize 4 broad nutritional categories of organisms: photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoautotrophs. Prokaryotes are represented in all 4 groups. Table 2. Photoautotrophs perform photosynthesis. They use light as their source of energy and carbon dioxide as their source of carbon. Like the photosynthetic eukaryotes, one group of photouautotrophic bacteria, the cyanobacteria, use chlorophyll as their key photosynthetic pigment and produce oxygen as a byproduct of non-cyclic electron transport. By contrast, the other photosynthetic bacteria use bacterial chlorophyll as their key photosynthetic pigment, and they do not release oxygen gas. Some of these photosynthesizers produce particles of pure sulfur instead because hydrogen sulfide H2S rather than H2O is their electron donor for photophosphorylation. Bacterial chlorophyll absorbs light at longer wavelengths than the chlorophyll used by all other photosynthesizing organisms does. As a result, bacteria using this pigment can grow in water beneath fairly dense layers of algae, using light at wavelengths that are not absorbed by the algae. Figure 2. Photoheterotrophs use light as their source of energy, but must obtain their carbon atoms from organic compounds made by other organisms. They use compounds such as carbohydrates, fatty acids, and alcohols as their organic food. The purple non-sulfur bacteria, among others, are photoheterotrophs. Chemoautotrophs obtain their energy by oxidizing inorganic substances, and they use some of that energy to fix carbon dioxide. Some chemoautotrophs use reactions identical to those of the typical photosynthetic cycle, see figure 8.3, but others use other pathways to fix carbon dioxide. Some bacteria oxidize ammonia or nitrite ions to form nitrate ions. Others oxidize hydrogen gas, hydrogen sulfide, sulfur, and other materials. Many archaea are chemoautotrophs. Deep-sea hydrothermal vent ecosystems are based on chemoautotropic prokaryotes that are incorporated into large communities of crabs, mollusks, and giant worms, all living at a depth of 2,500 meters below any hint of light from the sun. These bacteria obtain energy by oxidizing hydrogen sulfide and other substances released in the near boiling water that flows from volcanic vents in the ocean floor. Finally, chemoautotrophs obtain both energy and carbon atoms from one or more complex organic compounds. Most known bacteria and archaea are chemoheterotrophs, as are all animals and fungi and many protists. Nitrogen and sulfur metabolism. Many prokaryotes face important parts of their metabolism on reactions involving nitrogen or sulfur. For example, some bacteria carry out respiratory electron transport without using oxygen as an electron acceptor. These organisms use oxidized inorganic ions such as nitrate, nitrite, or sulfate as electron acceptors. Examples include the denitrifiers, bacteria that release nitrogen to the atmosphere as nitrogen gas N2. These normally aerobic bacteria, mostly species of the genera Bacillus and Pseudomonas, use nitrate NO3 as an electron acceptor in place of oxygen if they are kept under anaerobic conditions. 2NO3 plus 10E plus 12H plus right pointing arrow N2 plus 6H2O. Nitrogen fixers convert atmospheric nitrogen gas into a chemical form usable by the nitrogen fixers themselves as well as by other organisms. They convert nitrogen gas to ammonia. N2 plus 6H right pointing arrow 2NH3. All organisms require nitrogen for their proteins, nucleic acids, and other important compounds. The vital process of nitrogen fixation is carried out by a wide variety of archaea and bacteria, including cyanobacteria, but by no other organisms. Ammonia is oxidized to nitrate in the soil and in seawater by channelotrophic bacteria called nitrifiers. Bacteria of two genera, Nitrosomonas and Nitrosococcus, convert ammonia to nitrite ions, NO2, and Nitrobacter oxidizes nitrite to nitrate, NO3. What do the nitrifiers get out of these reactions? Their chemosynthesis is powered by the energy released by the oxidation of ammonia or nitrite. For example, by passing the electrons from nitrite through an electron transport chain, Nitrobacter can make ATP, and using some of this ATP, it can also make NADH. With this ATP and NADH, the bacterium can convert CO2 and H2O to glucose. Numerous bacteria base their metabolism on the modification of sulfur-containing ions and compounds in their environments. As examples, we have already mentioned the photoautotropic bacteria and channelotrophic archaea that use H2S as an electron donor in place of H2O. Such uses of nitrogen and sulfur have environmental implications, as we'll see in the next section. Prokaryotes in their environments. Prokaryotes live in and exploit all sorts of environments and are part of all ecosystems. In the following pages, we'll examine the roles of prokaryotes that live in soils, in water, and even in other living organisms, where they may exist in a neutral, benevolent, or parasitic relationship with their host tissues. Prokaryotes are important players in element cycling. Animals depend on photosynthetic plants and microorganisms for their food, directly or indirectly. But plants depend on other organisms, prokaryotes, for their own nutrition. The extended diversity of life on Earth would not be possible without nitrogen fixation by prokaryotes. Nitrifiers are crucial to the biosphere because they convert the products of nitrogen fixation into nitrate ions, the form of nitrogen most easily used by many plants. Plants, in turn, are the source of nitrogen compounds for animals and fungi. Denitrifiers also play a key role in keeping the nitrogen cycle going. Without denitrifiers, which convert nitrate ions back into nitrogen gas, all forms of nitrogen would leach from the soil and end up in lakes and oceans, making life on land impossible. Other prokaryotes contribute to a similar cycle of sulfur. Prokaryotes, along with fungi, return tremendous quantities of organic carbon to the atmosphere as carbon dioxide. In the ancient past, the cyanobacteria had an equally dramatic effect on life. Their photosynthesis generated oxygen, converting Earth from an anaerobic to an aerobic environment. The result was the wholesale loss of obligate anaerobic species that could not tolerate the O2 generated by the cyanobacteria. Only those anaerobes that were able to colonize environments that remained anaerobic survived. However, this transformation to aerobic environments made possible the evolution of cellular respiration and the subsequent explosion of eukaryotic life. What other roles do prokaryotes play in the biosphere? Archaea help stave off global warming. A time bomb lies deep under the ocean floor. Some 10 trillion tons of methane, potentially an overwhelming source of greenhouse gas, are located there. Will this methane escape to the atmosphere, hastening global warming? What will prevent such an escape is the presence of legions of archaea, also lying below the bottom of the seas. As methane rises from its deposits, it is metabolized by these archaea, with the result that virtually none of the methane even gets as far as the deepest waters of the ocean. Thus, these archaea play a crucial role in stabilizing the planetary environment. Prokaryotes live on in other organisms. Prokaryotes work together with eukaryotes in many ways. In fact, mitochondria and chloroplasts are descended from what were once free living bacteria. Much later in evolutionary history, some plants became associated with bacteria to form cooperative nitrogen-fixing nodules on their roots. The tsetse fly, which transmits sleeping sickness by transferring trichanosomes, microscopic protists described in the next chapter, from one person to another, enjoys a profitable association with the bacterium Wigglesworthia glossinidia. Biologists who decoded the genome of W. glossinidia in 2002 were surprised to learn that the bacterium's tiny genome contains almost nothing but the genes needed for basic metabolism and DNA replication, and 62 genes for making 10B vitamins and other nutritional factors. Without the vitamins provided by the bacterium, the tsetse fly cannot reproduce. The bacteria, living inside the fly cells, are in effect vitamin pills. Researchers are now trying to determine whether an attack on W. glossinidia may succeed in combating sleeping sickness, where more obvious direct attacks on tsetse flies or the trichanosomes have failed. Many animals, including humans, harbor a variety of bacteria and archaea in their digestive tracts. Cows depend on prokaryotes to perform important steps in digestion. Like most animals, cows cannot produce cellulase, the enzyme needed to start the digestion of the cellulose that makes up the bulk of their plant food. However, bacteria living in a special section of the gut, called the rumen, produce enough cellulose to process the cow's daily diet. Humans use some of the metabolic products, especially vitamins B12 and K, a bacteria living in the large intestine. We are heavily populated, inside and out, by bacteria. Although very few of them are agents of disease, popular notions of bacteria as germs arouse our curiosity about those few. Let's briefly consider the roles of some bacteria as pathogens. A small minority of bacteria are pathogens. The late 19th century was a productive era in the history of medicine, a time during which bacteriologists, chemists, and physicians proved that many diseases are caused by microbial agents. During this time, the German physician Robert Koch laid down a set of 4 rules for establishing that a particular microorganism causes a particular disease. 1. The microorganism is always found in individuals with the disease. 2. The microorganism can be taken from the host and grown in pure culture. 3. A sample of the culture produces the disease when injected into a new, healthy host. 4. The newly infected host yields a new, pure culture of microorganisms identical to those obtained in the second step. These rules, called Koch's postulates, were very important in a time when it was not widely accepted that microorganisms cause disease. Today, medical science makes use of other, more powerful diagnostic tools. However, one important step in establishing that the coronavirus was the causal agent of SARS, severe acute respiratory syndrome, a disease that first appeared in 2003, was the satisfaction of Koch's postulates. Only a tiny percentage of all prokaryotes are pathogens, disease-producing organisms, and of those that are known, all are in the domain bacteria. For an organism to be a successful pathogen, it must overcome several hurdles. It must arrive at the body surface of a potential host. It must enter the host's body. It must evade the host's defenses. It must multiply inside the host. It must damage the host to meet the definition of a pathogen. It must infect a new host. Failure to overcome any of these hurdles ends the reproductive career of a pathogenic organism. However, in spite of the many defenses available to potential hosts, some bacteria are very successful pathogens. For the host, the consequences of a bacterial infection depend on several factors. One is the invasiveness of the pathogen, its ability to multiply within the body of the host. Another is its toxicity, its ability to produce chemical substances, toxins that are harmful to the tissues of the host. Corynebacterium diphtheriae, the agent that causes diphtheria, has low invasiveness and multiplies only in the throat, but its toxicity is so great that the entire body is affected. In contrast, Bacillus anthracis, which causes anthrax, a disease primarily of cattle and sheep, but also sometimes fatal in humans, has low toxicity but an invasiveness so great that the entire bloodstream ultimately teems with the bacteria. There are 2 general types of bacterial toxins, exotoxins and endotoxins. Endotoxins are released when certain gram-negative bacteria grow or lice burst. These toxins are lipopolysaccharides, complexes consisting of a polysaccharide and a lipid component that form part of the outer bacterial membrane. Endotoxins are rarely fatal. They normally cause fever, vomiting, and diarrhea. Among the endotoxin producers are some strains of Salmonella and Escherichia. Exotoxins are usually soluble proteins released by living, multiplying bacteria, and they may travel throughout the host's body. They are highly toxic, often fatal, to the host, but do not produce fevers. Exotoxin-induced human diseases include tetanus, from Clostridium to tiny botulism, from Clostridium botulinum cholera, from Vibrio cholerae, and plague from Yersinia pestis. Anthrax results from free exotoxins produced by Bacillus anthracis. Remember that in spite of our frequent mention of human pathogens, only a small minority of the known prokaryotic species are pathogenic. Many more species play positive roles in our lives and in the biosphere. We make direct use of many bacteria and a few archaea in such diverse applications as cheese production, sewage treatment, and the industrial production of an amazing variety of antibiotics, vitamins, organic solvents, and other chemicals. Pathogenic bacteria are often surprisingly difficult to combat, even with today's arsenal of antibiotics. One source of difficulty is the ability of prokaryotes to form resistant films. Prokaryotes may form biofilms. Many unicellular microorganisms, prokaryotes in particular, tend to form dense films called biofilms rather than existing as clouds of individual cells. Upon contacting a solid surface, the cells lay down a gel-like polysaccharide matrix that then traps other bacteria, forming a biofilm. Once a biofilm forms, it is difficult to kill the cells. Pathogenic bacteria are hard for the immune system and modern medicine to combat once they form a biofilm. For example, the film may be impermeable to antibiotics. Biofilms often include a mixture of bacterial species. The biofilm with which you are most likely to be familiar is dental plaque, the coating of bacteria and hard matrix that forms between and on your teeth unless you do a good job of flossing and brushing. Biofilms form on contact lenses, on hip replacements, and on just about any available surface. Other biofilms foul metal pipes and cause corrosion, a major problem in steam-driven electricity generation plants. Biofilms are the object of much current research. For example, some biologists are studying the chemical signals used by bacteria and biofilms to communicate with one another. By blocking the signals that lead to the production of the matrix polysaccharides, they may be able to prevent biofilms from forming. Prokaryote phylogeny and diversity. The prokaryotes comprise a diverse array of microscopic organisms. To explore their diversity, let's first consider how they are classified and some of the difficulties involved in doing so. The nucleotide sequences of prokaryotes reveal their evolutionary relationships. Why do biologists want to classify bacteria and archaea? There are three primary motivations for classification schemes: to identify unknown organisms, to reveal evolutionary relationships, and to provide universal names. Scientists and medical technologists must be able to identify bacteria quickly and accurately. When the bacteria are pathogenic, lives may depend on it. Until recently, taxonomists based their classification schemes for the prokaryotes on readily observable phenotypic characters such as color, motility, nutritional requirements, antibiotic sensitivity, and reaction to the Gram stain. Although such schemes have facilitated the identification of prokaryotes, they have not provided insights into how these organisms evolved, a question of great interest to microbiologists and to all students of evolution. The prokaryotes and the protists have long presented major challenges to those who attempted phylogenetic classifications. Only recently have sysmatists had the right tools for tackling this task. Analyses of the nucleotide sequences of ribosomal RNA have provided us with the first apparently reliable measures of evolutionary distance among taxonomic groups. Ribosomal RNA, rRNA, is particularly useful for evolutionary studies of living organisms for several reasons. rRNA is evolutionarily ancient. No living organism lacks rRNA. rRNA plays the same role in translation in all organisms. rRNA has evolved slowly enough that sequence similarities between groups of organisms are easily found. Let's look at just one approach to the use of rRNA for studying evolutionary relationships. Comparisons of rRNAs from a great many organisms reveal recognizable short base sequences that are characteristic of particular taxonomic groups. These signature sequences, approximately 6 to 14 bases long, appear at the same approximate positions in rRNAs from related groups. For example, the signature sequence AACUUAAG occurs about 910 bases from one end of the small subunit of ribosomes in 100% of the archaea and eukarya tested, but in none of the bacteria tested. Several signature sequences distinguish each of the three domains. Similarly, the major groups within the bacteria and archaea possess unique signature sequences. These data sound promising, but things aren't as simple as we might wish. When biologists examined other genes and RNAs, contradictions began to appear and new questions arose. Analyses of different nucleotide sequences suggested different phylogenetic patterns. How could such a situation have arisen? Lateral gene transfer muddied the phylogenetic waters. It is now clear that, from early in evolution to the present day, genes have been moving among prokaryotic species by lateral gene transfer. As we have seen, a gene from one species can become incorporated into the genome of another. Mechanisms of lateral gene transfer include transfer by plasmids and viruses and uptake of DNA by transformation. Such transfers are well documented, not just between bacterial species or archaeal species, but also across the boundaries between bacteria and archaea and between prokaryotes and eukaryotes. A gene that has been transferred will be inherited by the recipient's progeny and in time will be recognized as part of the normal genome of the descendants. Biologists are still assessing the extent of lateral gene transfer among prokaryotes and its implications for phylogeny, especially at the early stages of evolution. Figure 3 is an overview of the major clades in the domains bacteria and archaea that we will discuss further in this chapter. This phylogeny is based on the evidence that is currently available, but keep in mind that a new picture is likely to emerge within the next decade, based on new nucleotide sequence data and new information about the currently understudied archaea. Mutations are a major source of prokaryotic variation. Assuming that the prokaryote groups we are about to describe do indeed represent clades, these groups are amazingly complex. A single lineage of bacteria or archaea may contain the most extraordinarily diverse species. On the other hand, a species in one group may be phenotypically almost indistinguishable from one or many species in another group. What are the sources of these phylogenetic patterns? Although prokaryotes can acquire new alleles by transformation, transduction, or conjugation, the most important sources of genetic variation in populations of prokaryotes are probably mutation and genetic drift. Mutations, especially recessive mutations, are slow to make their presence felt in populations of humans and other diploid organisms. In contrast, a mutation in a prokaryote, which is haploid, has immediate consequences for that organism. If it is not lethal, it will be transmitted to and expressed in the organism's daughter cells, and in their daughter cells, and so on. Thus, a beneficial mutant allele spreads rapidly. The rapid multiplication of many prokaryotes, coupled with mutation, natural selection, and genetic drift, allows rapid phenotypic changes within their populations. Important changes, such as loss of sensitivity to an antibiotic, can occur over broad geographic areas in just a few years. Think how many significant metabolic changes can occur over even modest time spans, let alone over the entire history of life on Earth. When we introduce the proteobacteria, the largest group of bacteria, you will see that its different subgroups have easily and rapidly adopted and abandoned metabolic pathways under selective pressure from their environments. The bacteria. The best studied prokaryotes are the bacteria. We will describe bacterial diversity using a currently popular classification scheme that enjoys considerable support from nucleotide sequence data. More than a dozen clades have been proposed under this scheme. We will describe just a few of them here. The higher order relationships among these groups of prokaryotes are not known. Some biologists describe them as kingdoms, some as subkingdoms, and others as phyla. Here, we simply call them groups. We'll pay the closest attention to 5 groups: the proteobacteria, cyanobacteria, spirochetes, chlamydias, and firmicutes. See figure 3. First, however, we'll mention one property that is shared by members of 3 other groups. Some bacteria are heat lovers. 3 of the bacterial groups that may have branched out earliest during bacterial evolution are all thermophiles, heat lovers, as are the most ancient of the archaea. This observation supports the hypothesis that the first living organisms were thermophiles that appeared in an environment much hotter than those that predominate today. The proteobacteria are a large and diverse group. By far the largest group of bacteria, in terms of numbers of described species, is the proteobacteria, sometimes referred to as the purple bacteria. Among the proteobacteria are many species of gram-negative, bacterial chlorophyll-containing, sulfur-using photoautotrophs. However, the proteobacteria also include dramatically diverse bacteria that bear no resemblance to those species in phenotype. The mitochondria of eukaryotes were derived from proteobacteria by endosymbiosis. No characteristic demonstrates the diversity of the proteobacteria more clearly than their metabolic pathways. Figure 4. The common ancestor of all the proteobacteria was probably a photoautotroph. Early in evolution, 2 groups of proteobacteria lost their ability to photosynthesize and have been chemoautotrophs ever since. The other 3 groups still have photoautotrophic members, but in each group, some evolutionary lines have abandoned photoautotrophy and taken up other modes of nutrition. There are chemoautotrophs and chemoautotrophs in all 3 groups. Why? One possibility is that each of the trends in figure 4 was an evolutionary response to selective pressures encountered as these bacteria colonized new habitats that presented new challenges and opportunities. Among the proteobacteria are some nitrogen-fixing genera, such as Rhizobium, another bacteria that contribute to the global nitrogen and sulfur cycles. E. coli, one of the most studied organisms on Earth, is a proteobacterium. So, too, are many of the most famous human pathogens, such as Yersinia pestis, Vibrio cholerae, and Salmonella typhimurium, all mentioned in our discussion of pathogens above. Fungi cause most plant diseases, and viruses cause others, but about 200 plant diseases are of bacterial origin. Crown gall, with its characteristic tumors, is one of the most striking. The causal agent of crown gall is Agrobacterium tumefaciens, which harbors a plasmid used in recombinant DNA studies as a vehicle for inserting genes into new plant hosts. Cyanobacteria are important photoautotrophs. Cyanobacteria, sometimes called blue-green bacteria because of their pigmentation, are photoautotrophs that require only water, nitrogen gas, oxygen, a few mineral elements, light, and carbon dioxide to survive. They use chlorophyll A for photosynthesis and release oxygen gas. Many species also fix nitrogen. Their photosynthesis was the basis of the oxygen revolution that transformed Earth's atmosphere. Cyanobacteria carry out the same type of photosynthesis that is characteristic of eukaryotic photosynthesizers. They contain elaborate and highly organized internal membrane systems called photosynthetic lamellae, or thylakoids. The chloroplasts of photosynthetic eukaryotes are derived from an endosymbiotic cyanobacterium. Cyanobacteria may live free as single cells or associate in colonies. Depending on the species and ungrowth conditions, colonies of cyanobacteria may range from flat sheets one cell thick to filaments to spherical balls of cells. Some filamentous colonies of cyanobacteria differentiate into 3 cell types: vegetative cells, spores, and heterocysts. Vegetative cells photosynthesize. Spores are resting cells that can eventually develop into new filaments, and heterocysts are cells specialized for nitrogen fixation. All of the known cyanobacteria with heterocysts fix nitrogen. Heterocysts also have a role in reproduction. When filaments break apart to reproduce, the heterocyst may serve as a breaking point. Spirochetes look like corkscrews. Spirochetes are gram-negative, mobile, chemotherapeutic bacteria characterized by unique structures called axial filaments, which are modified flagella running through the peroplasm. The cell body is a long cylinder coiled into a spiral. The axial filaments begin at either end of the cell and overlap in the middle, and there are typical basal bodies where they are attached to the cell wall. The basal bodies rotate, as they do in other prokaryotic flagella. Many spirochetes live in humans as parasites. A few are pathogens, including those that cause syphilis and Lyme disease. Others live free in mud or water. Chlamydias are extremely small. Chlamydias are among the smallest bacteria, 0.2-1.5 micrometers in diameter. They can live only as parasites within the cells of other organisms. These tiny gram-negative cocci are unique prokaryotes because of their complex life cycle, which involves two different forms of cells, elementary bodies and reticulate bodies. In humans, various strains of chlamydia cause eye infections, especially trachoma, sexually transmitted diseases, and some forms of pneumonia. Most firmicutes are gram-positive. The firmicutes are sometimes referred to as the gram-positive bacteria, but some firmicutes are gram-negative, and some have no cell wall at all. Nonetheless, the firmicutes constitute a clade. Some firmicutes produce endospores, heat-resistant resting structures, when a key nutrient such as nitrogen or carbon becomes scarce. The bacterium replicates its DNA and encapsulates one copy, along with some of its cytoplasm, in a tough cell wall heavily thickened with peptidoglycan and surrounded by a spore coat. The parent cell then breaks down, releasing the endospore. Endospore production is not a reproductive process. The endospore merely replaces the parent cell. The endospore, however, can survive harsh environmental conditions that would kill the parent cell, such as high or low temperatures or drought, because it is dormant. Its normal activity is suspended. Later, if it encounters favorable conditions, the endospore becomes metabolically active and divides, forming new cells like the parent. Some endospores can be reactivated after more than 1,000 years of dormancy. There are credible claims of reactivation of bacillus endospores after millions of years, and even one claim of uncertain validity of more than a billion years. Members of this endospore forming group of firmicutes include the many species of Clostridium and Bacillus. The toxins produced by C. botulinum are among the most poisonous ever discovered. The lethal dose for humans is about 1 millionth of a gram, 1 microgram. B. anthracis, as noted above, is the anthrax pathogen. The genus Staphylococcus, the Staphylococci, includes firmicutes that are abundant on the human body surface. They are responsible for boils and many other skin problems. S. aureus is the best known human pathogen in this genus. It is found in 20 to 40% of normal adults and in 50 to 70% of hospitalized adults. It can cause respiratory, intestinal, and wound infections in addition to skin diseases. Actinomycetes are firmicutes that develop an elaborate system of filaments. These bacteria closely resemble the filamentous growth habit of fungi at a reduced scale. Some actinomycetes reproduce by forming chains of spores at the tips of the filaments. In species that do not form spores, the branch, filamentous growth ceases and the structure breaks up into typical cocci or bacilli, which then reproduce by fission. The actinomycetes include several medically important bacteria. Mycobacterium tuberculosis causes tuberculosis. Streptomyces produces streptomycin as well as hundreds of other antibiotics. We derive most of our antibiotics from members of the actinomycetes. Another interest